Today in this class, I am explain about genetically modified foods. Introduction. Genetically modified foods are foods produced from organisms that have had changes introduced into their DNA using the methods of genetic engineering as opposed to traditional crossbreeding. In the US, the Department of Agriculture and the Food and Drug Administration favor the use of the term genetic engineering over genetic modification as being more precise. The USDA defines genetic modification to include genetic engineering or other more traditional methods. According to the World Health Organization, foods produced from or using genetically modified organisms are often referred to as genetically modified foods. Advances in plant biotechnology have made it possible to identify and modify genes controlling specific characters. Nowadays, scientists can transfer genes from one organism to another unrelated organisms, producing what is now known as genetically modified organism or transgenic animals or plants. Any food produced this way is called genetically modified foods, that is, foods obtained by adding or deleted gene sequence. More technically, genetically engineered foods are defined as food produced from plants, animals, and microbes that have at their genetic code modified by the selective introduction of specific DNA segments that called as genetically modified foods. And this process allows the organism to acquire a desired trait such as pest protection, herbicide resistance, or improved nutritional qualities. The enhancement of desired traits has traditionally been undertaken through breeding, but conventional plant breeding methods can be very time consuming and are often not very accurate. And the peculiar features which could define genetically modified foods are food that contain an added gene sequence and food that have a deleted gene sequence, and the animal products from animal feed, genetically modified feed, and products produced by genetically modified organisms. Next one, history of genetically modified foods. In 1960, a lot of breakthrough were recorded in the field of genetics. It was proven that this new knowledge had the potential to revolutionize food production, thus creating huge benefits of the world. By 1972, another scientific breakthrough was recorded by Paul Berg, who joined together DNA from two different organisms to create the first recombinant DNA molecule. This breakthrough was followed by a pioneer study in which Stanley Cohen and Robert Boyer inserted DNA from an African clawed toad into the Escherichia coli bacterium. Shortly after then, some companies realized that this fledgling technology could open up new highly profitable market. And next one, types of genetically modified foods. First type, first generation crops. They have enhanced input traits such as herbicide tolerance, better insect resistance, and better tolerance to environmental stress. And the ensuing crops are not significantly different from the traditionally grown crops in terms of appearance, taste, and nutrition. For example, of such crops are herbicide resistant soybean, insect resistant maize, and herbicide and insect resistant potatoes. And second one is second generation crops. They have new traits to increase their benefit to consumers, such as increased levels of proteins, modified or healthier fats, modified carbohydrates, and increased flavor or increased micronutrients. For example, of such crops includes rice with a higher level of beta carotene and tomatoes with higher levels of carotenoids, maize with increased vitamin C, and soybean with improved amino acid compositions, and potatoes with higher calcium content. And third one is third generation crops. These genetically modified foods are in the research pipeline. These plants may have traits that can provide increased ability to resist abiotic stress such as drought, increased temperature, or saline soils. Other traits may provide health benefits. Another objective may be to create pharmaplants to help produce active pharmaceutical products. And in 2009, the US Food and Drug Department approved the license for a recombinant antithrombin for prevention of food blood clots in patients with hereditary antithrombin deficiency. And the recombinant antithrombin is the first human biologic drug derived from the milk of goats that have been genetically engineered to produce human antithrombin in their milk. And next topic is genetically modified food techniques. There are three main types of genetically modified food techniques. First one, inserting genes, that is gene shifting. Genes are determined by different DNA sequences. When the isolated gene is inserted into a plant, it becomes part of the plant's and gene and work with its own function. This method can increase or improve the plant such as resistance to insect, which increased the yield of food afterwards. And second one is removing genes, that is gene silencing. The function is reduced or stopped through genetic modification. For example, the function of virus which cause dried and spot of tomato is reduced by removing parts of the genes. Thus, the virus cannot be reproduced and tomato can grow healthily. And third one is changing the process of catabolism, that is genes splicing. Food can be enhanced by changing the process of catabolism, such as controlling the percentage of starch of glutinous rice, and is also include controlling the taste, mass, color, and usefulness of foods. And next one is general genetically modified food process. First one, isolation of gene of interest. Second one, insertion of the genes into the transfer vector. Next, third one is plant transformation. Fourth one, selection of modified plant cell. And fifth one, regeneration into whole plants via tissue culture. And sixth one, verification of transformation and characterization of the inserted DNA fragment. And seventh one, testing of plant performance. And eighth one, safety assessment. First one, general genetically modified food process. First one, isolation of the genes of interest. A chromosome is used to identify the genes responsible for the desired trait in the organism. And second one is insertion of the genes into a transfer vector. The desired trait is put into the plasmids. And third one is plant transformation. The plasmid contain inside agrobacterium tumefaciens cells transfers the plasmids and new gene into the plant's chromosome. And fourth one, selection of the modified plant cell. Selectable marker gene are used to favor the growth of the cells containing the trait as opposed to the non-transformed cells. And fifth one, regeneration into the whole plant via tissue culture. In this step, they take explant on media containing nutrients that spark develop of cells that form plantlets that once rooted are put in pots controlled in environmental condition. And sixth one, verification of transformation and characterization of the inserted DNA fragment. Tests are done to determine the number of copies if intact and doesn't interfere with other genes. These tests are done to see if the gene is functional. And seventh one, testing of the plant performance. Here we see or test and see if the resulted plant grown in a greenhouse has acquired the favored trait and if it has any unwanted characteristics. And last one, safety assessment. Now more tests carried out to see plants performance, environmental safety assessment and other safety assessment. Next one is common genetically modified foods. First one, soya bean, corn and sugar beet. This is resistance to glyphosate by inserting herbicide resistance gene. Second one, cotton seed oil. It is by inserting pest resistance Bt crystal protein gene. And third one is tomato. This is by removing the gene that codes for polygalactoronase responsible for softening of fruits after harvesting. The fourth one is potatoes. Amylopectin rich variety by switching off of GBSS, that is granule bound starch synthase gene responsible for amylose production. And next one is canola. Canola is with high oleic acid content by adding new gene. Next last one, rice with high vitamin A by inserting gene from daffodils. Next one is more specific example of genetically modified foods. First one, golden rice. It was created by Ingo Patricus. Golden rice is a variety of rice produced through genetic modification to biosynthesize the precursor of beta carotene in the edible part of rice, that is in the part of endosperm. More, more than 120 million children in the world suffer from vitamin A deficiency. Golden rice has the potential to help prevent the 1 to 2 million deaths each year caused by a deficiency in this vitamin. Golden rice was created by incorporating rice with 2 beta carotene biosynthesis gene, that is PSY, that is phytone synthase, and second one LYC, that is lycopene cyclase gene. And second one is cold treatment tomatoes. Scientists have created a frost resistant tomato plant by adding an anti-freezing gene from the cold water fish to it. The anti-freezing genes come from the cold water flounder, a fish that can survive in very cold conditions. The flounder has a gene to make chemical antifreeze. This is removed from the antifreeze DNA and is joined on a piece of DNA called a plasmid. This hybrid DNA, which is a combination of DNA from two different sources, is known as recombinant DNA. And next one is vitamin rich tomatoes. The agrobacterium naturally infects plants by causing various diseases. By replacing that gene with desired ones results in the new genetic makeup with advantageous traits. The bright orange color of carrots come from beta carotene, which work as the precursor for the synthesis of vitamin A in our body. So by inserting this color gene into tomato, enhance its appearance as well as its vitamin A level to desired level. And next one, protein enriched potatoes for NASA. Potato is non-cereal food crop limit in the amount of lysine, tyrosine, methionine and cysteine to provide sufficient protein and amino acids to astronauts with their complete nutritional requirements. A tuber species protein, amaranth seed, albin has been used to transform potatoes. The albumin protein has a well balanced amino acid profile. In fact, its amino acid composition exceeds values recommended by the World Health Organization for a nutritionally rich protein. This protein was used due to its non-allergenicity in its purified form. When the albumin gene was inserted into a potato, 2.5 to 4-fold increase in lysine, tyrosine, methionine, and cysteine content, and 35 to 45% increases in total protein content was reported in transgenic tuber. And next one is BT soybean. The two target insects for insect resistance transgenic soybeans are the velvet bean caterpillar and the soybean looper. These pests feed on the leaves of the soybean plant and can severely limit yield. Velvet bean caterpillar population can reach damaging level rapidly. Many producers in areas where velvet bean caterpillar is a significant problem apply a preventive treatment of dimilin when plants are in full bloom. Scientists have incorporated Bacillus thuringiensis gene into soybean, which has insecticidal protein that maintains the yield of the crop. Bacillus thuringiensis is a ubiquitous soil bacterium is the source of the gene for insect resistance. Next one, genetically modified corn. Corn has been deliberately genetically modified to have agronomically desirable traits. The traits that have been engineered into corn include resistance to herbicides and resistance to insect pests, the latter being achieved by incorporation of a gene that codes for the Bacillus thuringiensis toxin. Corn varieties resistant to glyphosate herbicides have been produced. Pioneer Hybrid has marketed corn hybrids with tolerance to imidazoline herbicide under trademark Clearfield. And next one is Roundup Ready soybean. The Roundup Ready soybean is a transgenic soybean that has been immunized to Roundup herbicide. Since the soybeans naturally trypsin inhibitors provides protection against pests, the only major problems in soybean farming was weeds, thus making soybean revolution. The glyphosate in the herbicide would inhibit the soybean plants which is involved in the maintenance of biosynthesis of aromatic metabolism and cause the plant to die along with the weeds for which the herbicide was meant. Next one is canola oil. The canola oil had a distinctive taste and the disagreeable greenish color due to the presence of chlorophyll. It also contains a high concentration of erucic acid. Experiments on animals have pointed to the possibility that erucic acid consumed in large quantities may cause heart damage. In non-toxin, the cultivar used produced commercial food grade canola oil was bred to contain less than 2% erucic acid levels that are not believed to cause harm in humans, and no ill health effects has been associated with consumption by humans of the genetically modified oil. Next one, papaya. Papaya cultivation is threatened by papaya ring spot virus, a disease that sharply lowers the fruit yield. The University of Hawaii developed a ring spot virus disease resistance papaya to do this. Certain viral genes encoding capsid proteins were transferred to the papaya genome. These viral capsid proteins elicit something similar to an immune response from the papaya plant. The first resistant papaya varieties were grown commercially in 1999 in Hawaii. These genetically modified papayas are approved for consumption both in US and Canada. With canola oil, rapeseed oil had a distinctive taste and disagreeable greenish color due to the presence of chlorophyll. It also contains a high concentration of erucic acid, and experiments on animals have pointed to the possibility that erucic acid consumed in large quantities may cause heart damage. A known toxin, the cultivar used to produce commercial food grade canola oil was bred to contain less than 2% erucic acid, and levels that are not believed to cause harm in humans, and no ill health effects has been associated with consumption by humans of genetically modified oil. And next one is, why produced genetically modified food? Traditionally combining the desirable genes Traditionally combining the desirable genes in one plant is tough task that utilize a longer time and so much attention involving crossing one plant to another plant of the same species or related species. From economical and agricultural standpoint, it is advantageous to grow crops that have higher yield or improved quality, pest or disease resistance or tolerance to heat, cold and drought. Desirable genes may provide means for plants to combat these conditions, and the development of transgenic technology allows useful genes from various living sources to be brought together in a relatively simple manner. Advantages of genetically modified food. First one, improve yield and more resistance to disease and less likely to damaged by insect, and tolerance to herbicides and better nutritional value and increased shelf life and improved in health and environment and better climate survival by increasing tolerance to drought, flood, and to allow the use of previously inhospitable land and higher crop yields and reduced farm cost and increased farm profit. That's about the genetically modified foods. Thank you.